In this video, I'll be tackling the principles of clinical trials in oncology. I'm going to be trying to demystify clinical trials for patients. I'm going to be discussing what is a clinical trial, what is clinical research, so that we can have evidence-based medicine, all with the intention to, of course, try and improve outcomes with patients, but also first do no harm. We're going to look at statistics and hypothesis testing at its basics so that we understand why and how clinical trials are designed. And we're going to speak specifically about biases, selection bias, physician and patient bias, and also talk about random variation. And we're going to talk about strategies to limit each of these problems, first with randomization and also looking at a placebo-controlled study. I'm going to introduce the different types of clinical trials, phase 1 studies, phase 2 studies, phase 3 studies, and take you through the timeline as an example of zolbetuximab for claudin 18.2 positive tumors, relevant now for gastroesophageal cancer in 2023. I'm also going to introduce the topic of fast track or accelerated approvals of the FDA for various therapies that bypass some of these traditional steps in clinical trials. And then finally, I'm going to talk about the logistics of studies, speaking to trial eligibility and also the costs of studies, the duration of the treatment on the studies, and where these studies are implemented. So first, why do we do what we do as a standard of care? Well, generally speaking, the strategies that we use at any given setting, whether it's curative or metastatic, is based on information that we've learned in the past, most often in the setting of a clinical trial. So for example, here we see in the first line setting, the first treatment used for newly diagnosed stage 4 or metastatic gastroesophageal cancer, we know historically that with no therapy intended to treat the cancer other than supportive measures, referred to as best supportive care, the median survival times, which as you see there, is not what any individual experience, but it is an estimate of the time point at which half of the patients are still alive and half the patients are not alive from the time of diagnosis, is about 3 to 6 months, depending on the study and the data set being evaluated. With our best chemotherapy treatments in the newly diagnosed setting, the median survival is around 10 to 12 months. And now with the introduction of specific biomarkers and their match targeted therapies, in those groups, it's a little bit higher, as I'll show you. But generally speaking, for a HER2 negative, PD-L1 negative, claudin negative tumor, the median survivals range around 10 to 12 months with our optimal therapies today. And we know from clinical trials that chemotherapy improves the median survival, as you can see, from around 3 to 6 months median to around 10 to 12 months median. But even more importantly, we've learned that when administered appropriately, it is palliative, meaning it improves the quality of life of patients by, for example, decreasing pain symptoms from the cancer, improving dysphagia, the problems with swallowing, etc. And again, we've learned all of this through clinical trials and their results that have been conducted over the last decades. At the time, those treatments were not considered standard, but because in the setting of a clinical trial, they improved upon best supportive care, they became a new standard approach at the time. And so now they are referred to as standard therapy. So what led us to implement first-line chemotherapy? Well, we have to go back several decades to the 90s when studies were being performed comparing chemotherapy of various types to best supportive care. And best supportive care really involves pain medications, IV fluids, antibiotics if necessary, anti-nausea medication, anything that can palliate the symptoms of the cancer but aren't treating the cancer. The next thing we learn from this figure is something called a hazard ratio. A hazard ratio is really looking at what is the comparison of the two groups and what is the outcome. So in this study, we're comparing chemotherapy to best supportive care. And if the hazard ratio is 0.667, what is that indicating? That's indicating that the therapy is better. And so another way to look at it is to say, if we improve survival from 6 months median to 9 months median, this would be a hazard ratio of 0.667. And so from the curve below, you see if the estimate of the outcome is on the line of 1, then there's no difference between the two different treatments being compared. If the estimate is to the left of 1, anywhere less than 1, then it would favor one of the arms, in this case, chemotherapy. If, on the other hand, the estimate was on this side, it would be higher than 1 and it would favor the other group, in this case, best supportive care. So this is how we would look at something called a forest plot. This is a forest plot. The next thing to understand is a 95% confidence interval. And so these bars range across the point estimate. And what this is indicating is that from this study and the characteristics of the results, we estimate that the benefit is at this value, a hazard ratio of 0.33, but we are 95% confident that it is within this boundary of 0.17 to 0.64. We're going to get to, in a little bit, the statistics around this and the uncertainty of the actual estimate and why we put 95% confidence intervals around that estimate. The next thing to understand is in this particular analysis, which is a meta-analysis, it's looking at 3 different studies that ran 3 different studies. The numbers of patients enrolled are here, and in each arm of the study, each group of the treatment arms. And we see each individual estimated hazard ratio. What a meta-analysis does is that it pools them all together to help with getting a more confident result. And so the hazard ratio here is an aggregate of each of the 3 weighted by the numbers of patients enrolled. So the highest enrolling study gets more weight. And ultimately, a more precise estimate of the hazard ratio and usually a more narrow range of the 95% confidence interval is obtained. And so the reason one would do a meta-analysis here is because each of these are relatively small studies, but together it's a little bit larger and we can be a little bit more confident with the result. And so this analysis, amongst many, looking at whether monotherapy chemotherapy compared to best supportive care was beneficial. And you can see that it was, and that in this particular analysis, the median survival of best supportive care was 4.3 months, and it was increased to 11 months with single agent monotherapy chemotherapy, which became a standard of care. The principles that we learn here from this is that as we increase the numbers being evaluated, we increase the confidence and the precision of that estimated hazard ratio. And in the setting of where the benefit is actually small, it actually increases the chances by increasing the numbers that we will find a statistically significant finding. And we'll get more to that in the next slides. The next question was, what if we do 2 chemotherapy drugs compared to 1? Is that better? And so, as you can see here, over the last decades, a whole slew of studies with various numbers of patients enrolled, looking at their forest plots of hazard ratios, you can see that some studies weren't showing much different. Some studies were showing benefit, but together, adding them all together, weighted by the numbers of patients enrolled, you can see that the conclusion was that there is a benefit of a hazard ratio of 0.84 with a 95% confidence interval of 0.79 to 0.89, and suggesting that there is a benefit, albeit modest, in improvement in median overall survival from 10.5 months to 11.6 months by adding a second therapy. So this is, again, speaking to the principle that the higher the numbers of patients being assessed, the more likely you will find a statistically significant benefit, even if it's smaller. And again, statistically significance refers to the fact that we feel like this was not due to random variation. And we'll get to that in a few slides. But suffice it to say, at this point, we can say that it is statistically significant, that point estimate of 0.84 benefit, if the upper limit of that confidence interval is less than 1. That is, that the 95% confidence interval upper boundary is less than 1. So why do we use 5-FU in platinum therapy as our first treatment? Well, this meta-analysis, along with others, supports that. And this is a study specifically looking at 5-FU as a control arm compared to two multi-drug regimens, one being 5-FU and cisplatin, and the other being 5-FU, adriamycin, and mitomycin C. This regimen is an outdated regimen, not used anymore. But you can see this is an example of a clinical trial that randomized patients to 3 possible treatments, the numbers of patients in each arm, and the results showing that there was a higher percent of patients deriving shrinkage or partial response or complete response compared to the other arms. It also showed that the progression-free survival, or in this curve, the percent in progression, was higher in the 5-FU monotherapy arm compared to those with the cisplatin and 5-FU arm. And over here, you see the survival was a little bit higher with 5-FU and cisplatin compared to 5-FU. Now, this wasn't statistically significant, but this gets to the fact that these are relatively small numbers. Weren't able to establish statistical significance, but in meta-analyses like this one, it is supported as a statistically significant improvement, albeit modest, over monotherapy. So this is what has established 2-drug chemotherapy as our preferred first-line therapy in the palliative stage 4 setting. So as you can see, though, always more work needs to be done to improve on what is considered standard at any given time. To prove a new therapy, it must be with a clinical trial, and this is the purpose of this presentation. But why? I alluded to earlier that as a medical physician, we take an oath, first do no harm. And so we would like to know if any therapy is going to be harmful, what those side effects might be, so we can describe those to patients so they're informed before we get started, and also whether it's effective. And if it is effective, how effective? What is the magnitude of benefit? So all of these things are important to be able to provide evidence-based medicine and to first do no harm, first described by Hippocrates long ago. So what is a clinical trial? A clinical trial is an experiment designed to answer any specific question about biomedical or behavioral interventions, including therapy, like new investigational drugs for cancer. These clinical trials are only conducted after approval of health authorities and ethics committees, something called an IRB, or Institutional Review Board, that review clinical trial protocols that are basically the rules of how the trial will be conducted. And they vet those, and they look at what are the potential risks and what are the potential benefits, and is this an ethical thing to be doing? It does require informed consent, which means we review all of the aspects of the clinical trial in a clinical trial consent form that is then signed by the patient. The next thing to understand is that clinical trials are sponsored. They're sponsored by various entities. One can be the government. The National Cancer Institute provides funding to run clinical trials through cooperative groups. An example of a cooperative group is the Alliance for Clinical Trials Group, which is a collection of various academic centers that come together to run studies. Other examples are ECOG, Eastern Cooperative Oncology Group, or SWOG, Southwestern Oncology Group, or NRG, the National Radiation Group. And so that is one form of a sponsor. Another sponsor could be an academic center, a principal investigator, an investigator physician at a center that comes up with a study, writes the protocol and consent form, gets funding from a source to be able to run it. And these studies are referred to as ISTs or IITs, investigator-initiated studies or trials. And then finally, probably the most common, for better or worse, is a commercial company or a pharmaceutical company sponsored study, where they are the ones that are sponsoring the study, they're designing the study. And I'll point out along the way as we go through examples of different studies where sometimes issues can arise in terms of biases and the way the trials are designed. So before we even get into clinical trials in patients, we usually have therapies that are being assessed, what is referred to as preclinically, before they get in the clinic, before they're administered to patients. And these are in laboratories across the world that can be, again, the academic center laboratories and government institution laboratories and pharmaceutical company laboratories, evaluating the biology of cancer and designing new therapies based on what is learned. These are often done in cancer cell lines that are obtained from patients and grown in the laboratory. They're also in animal models that can be used using cell lines to grow in the animal models, or in animal models that have initiated cancer in their cells and then treated. And then after several years of this analysis, the promising agents can then be brought forward into human studies. The first in human studies of a new drug are called phase 1 studies, and they're typically smaller, usually, historically at least, less than 20 patients. And classically, they would be run at one treatment center. In the last years, they've been sort of expanded, and they're usually done at multiple centers at the same time. This is a function of pharmaceutical companies trying to get information as quickly as possible. And so by opening it at multiple centers, then patients can be getting enrolled simultaneously at different centers and facilitate the accrual and accelerate the completion of the studies. And the primary intention of these studies is to really determine the drug's safety, what its dosing schedule should be, how often, and whether this drug is tolerable. The way this is done is that usually patients are usually enrolled at 3 patients at a time at a given dose, a starting dose, and they're followed for usually a minimum of about 1 month to show that it's safe before the dose can be titrated upwards to the next dose level that's pre-specified in the protocol. And 3 more patients would be enrolled at that new dose level. And this would be continued typically until there's a maximum tolerated dose or a maximum biologically effective dose that even if the drug doesn't cause side effects, we're not going to just keep going up indefinitely. There will be sort of a ceiling at which we feel like this level should be an effective dose based on what we understand about this drug. And so the phase 1 study will be conducted in order to pinpoint and solidify what that dosing schedule will be, get some understanding of what the safety will be, and also early efficacy. But ultimately, this will then be used, this dose will be used in the phase 2 study that would follow the phase 1 study upon completion. This is an important point to make here, is that in the phase 1 setting, many patients may be experiencing this, say now, for example, with CAR T cells studies, where they're waiting to get onto a study, but they're in this waiting period of where the study is actually closed to accrual while previous patients are being followed to look at safety and what the dosing should be. And so we're waiting until a new cohort opens up. And so this is why you're waiting, is because some patients have been enrolled and now we're being followed before we will enroll more patients to ensure safety. So one might experience that. And again, an example of that right now is with CAR T studies that are sort of sought after and desired, but they're not always open. Even though the study's open at a center, the cohort is not enrolling. And so we sort of have to wait until that reopens. For the patient, of course, both the patient and the physician who enroll the patient on a study, we hope that they derive benefit from the drug at any given dose that it's being given. But it should be understood that that's not the primary endpoint of a phase 1 study. It is a secondary endpoint, looking at what are the outcomes and what are the clinical benefit of the patient. So usually, especially if this is a first in human therapy, therefore this type of a phase one study is usually reserved until after standard therapeutic options are used and no longer working, or studies that are in later stage of development, phase two or three, as we'll get to. But ultimately, there comes a point where this type of a phase one study is really the only other option other than best supportive care. And if a patient is willing and otherwise eligible, that we'll get to in terms of eligibility criteria, then this would be a reasonable thing to consider. Something to understand, though, is that this may not necessarily be a first in human study. A phase one study may just be the first time that two other agents that have already been through all the phases of study and already may be commercially available for treatment, but it's the first time they're being combined together. And so if that's the case, then it would need to be done in a phase one study to show safety of this combination and to chronicle sort of what are the side effect profiles now that they're being added together. And do we need to alter the doses, for example, to make them better tolerated, and to find new dose levels if they are combined together? And so we're back to the drawing board with a phase one study. An example of this, we ran a study of 4 already approved chemotherapy drugs, but it was the first time they've ever been combined together and given at the same time. And so this was an example of a study that was being done in the first line newly diagnosed setting in the stage 4 setting, but because it was a combination of drugs that have never been combined together simultaneously, it was called a phase 1 study. So regardless, when a safe and tolerable dose is established in a phase 1 study, classically, the new investigational drug would proceed to a phase 2 study. Phase 2 studies typically range from anywhere between 60 to around 200 patients in size. And for example, a smaller pilot study just to explore a little bit further to build upon what was learned from the phase 1 study, 60 more patients might be enrolled without any randomization to just get a little bit more patients to really get a better understanding of what the clinical benefit might be. And so without randomization, it's referred to as a phase 2A study. On the other hand, if the drug is showing a lot of promise or it has shown promise in a phase 2A study, then the next step might be to do a phase 2B study, a larger but randomized phase 2 study that is a little bit larger with, say, 100 patients in each arm. These types of studies are usually multi-center because in order to accrue this many patients, we need to have more centers accruing to the study to get the numbers of patients in a reasonable amount of time. Sometimes there are even international studies that are run globally in different countries. The numbers of patients we're going to get to, how does it decided how many patients are going to be enrolled to a given study? And we'll get to that in terms of the statistic calculation. Something important to understand is that once the target number of patients is met, and in that many patients have been enrolled to the study, the trial will then close to accrual and will not allow any other patients to enroll to that particular study. Now, the primary reason for a phase 2 study, or the primary endpoint it would be referred to as, is efficacy, the outcome. How does this improve upon what we might expect otherwise in the standard of care of whatever the setting is that this study is running? And primary endpoints can be overall survival. They can be progression-free survival, the time that it takes before the therapy stops working, or it can be the response rate, how many patients experience shrinkage of their cancer of 30% or more from what it started as. And so whichever one is decided to be the primary endpoint, often these other endpoints are referred to as secondary endpoints. In addition to these clinical efficacy endpoints, always secondary endpoints included are the tolerability and the side effect profile of the therapy to build on what was learned from the phase 1 study and earlier phase studies to really get a sense of what the potential benefits and what the potential risks of any given therapy are. So phase 2 studies are not usually what would be referred to as practice changing. They're not registrational studies. They're not seeking FDA approval for the therapy. Rather, they're serving as a checkpoint, as an intermediate before running such a study called a phase 3 study to ensure that there's enough evidence that this drug will be safe enough and beneficial enough to proceed to a phase 3 study. Trials are expensive. They take a lot of time, several years, as I'll show you with the zolbetuximab example. And if we could learn earlier from a phase 2 study that's smaller and therefore run a little bit quicker, that the drug is not effective or not safe, or both, then it's in everyone's interest, and most importantly, patients, that we're not enrolling patients to study that we could have learned earlier that it wasn't going to be an effective therapy. So it's a checkpoint to exclude drugs that are not worthy enough to proceed to registrational purposes. And so that said, sometimes a company may skip a phase 2 study and just go from phase 1 that has established safety and some preliminary side effect profile and a dose, and go directly to a larger registrational phase 3. This is often a discussion of companies based on this limited data that they have from small sample sizes, and racing against other companies looking at similar agents, for example, and basically trying to make a decision of whether they should or should not just go for it in the phase 3 setting. And so there will be examples that you have a phase 3 study which didn't have a phase 2 study and just jumped from phase 1 to phase 3. Regardless, if a phase 2 study is positive, or if the phase 1 study data are really exciting, then the sponsor will decide to proceed to a phase 3 study. Phase 3 studies are much larger and typically range anywhere between 200 to even more than 1,000 patients. I'll show you some examples of that. And they are usually randomized with an appropriate control arm, randomized to whatever the standard of care is in that setting of where the phase 3 study is being run. And sponsors, again, let's say, for example, a pharmaceutical company, will discuss with the regulatory agencies like the FDA in the United States, or the European equivalent of that, the EMA, prior to finalizing their protocol, so that, you know, when all is said and done and the study is completed, then it was predefined that if this study is positive, then this would lead to registration. These studies are, again, multi-center given the numbers of patients, and very often they're global and international studies where multiple centers are enrolling to this study. For example, each center may only accrue 2 to 3 or 4 patients, but when you have so many centers, you can get to these large numbers for the whole study. Again, so if the study is positive in the end, showing that there is a true benefit compared to the standard of care, then this new drug would be eventually FDA approved after the data are all reviewed, and they would be approved for the specific indication that the study was run and become a new standard of care. The FDA does take some time to review the data even after it's reported, and so these drugs aren't immediately available, and it may take some time, months usually, and hopefully sometimes years, to approve a drug and make it actually available for patients. And an example of that presently in April of 2023 is zolbetuximab, which has 2 positive phase 3 studies called Spotlight and Glow, showing overall survival benefit. And they were just recently presented in the last few months. But we are waiting now for approval so that we can actually get access to the therapy. So I'm going to take you through the timeline of the zolbetuximab story just to show you how long it may take for a drug to go from inception to actually being available for patients routinely for therapy. So the first publication of a preclinical study in animal models and cell lines is from 2008 for this drug. It used to be called IMAB 362. When drugs are being developed, they're given a number, and ultimately, if they get through all of the different stages and they're showing that they're likely to become a therapy that's approved, then they'll be given a name. Again, remember from the targeted therapies video that drugs that are antibodies end in AB and monoclonal antibody. So you can bet that this study being published in 2008 was showing that the drug itself was being designed years even before that, before the final publication of this preclinical study was available. Regardless, then the first presentation of the first in human studies, phase 1 and 2 studies, was at an abstract at the annual American Society of Clinical Oncology meeting in 2013. Then the phase 2b study, the randomized study, which looked at standard chemotherapy with or without the antibody, was first reported orally in 2016 at the GI ASCO meeting. And then publication of this previous phase 1 study came out in 2018. And then the first publication of the phase 2a study, a non-randomized study looking at larger numbers of patients with gastroesophageal cancer called the MONO study for second line and higher, was published in 2019. Now, in this case, there was a little bit of delay before these publications came out because the antibody rights was changed from one company, a German company, to Astellas, which is now the company developing it further. And so there was a delay in that transition that led to a little bit more time in between these presentations and their publications. And then the publication of the randomized phase 2b FAST study was actually published in 2021, just recently. And then the first presentation of the phase 3 study called Spotlight was in January of 2023 at GI ASCO. And then the GLOW study was reported just last month in March of 2023. And so now, as I mentioned, we're waiting now for the final approval of the FDA and other regulatory agencies across the world to approve this. And we're hoping that we see an approval by the summer of 2023. So the point of showing you this is that this is a classic development of a therapy starting from preclinical, going through phase 1, 2, and then 3 studies, and that this can take, 15 plus years for this to really get to the clinic. Some other points to note, though, are that patients who enrolled on these studies early got access to this therapy, and in their case, probably helped them a little bit. And so that said, as I'll also show you, this is a success story, but there are a number of examples of drugs that get through these various stages and ultimately don't show benefit and ultimately are not approved. And so we just have to understand that as many drugs get through this process and show benefit, many more fail and fall out at these various steps and don't show the benefit. So why so long to prove a therapy, and why do we randomize? Randomization to compare to a standard control is required to control for various known and unknown factors that we call confounders that may affect the outcome. For example, survival. So we know, for example, patients that are younger will generally have a better survival compared to those who are older, because typically those who are older have other medical problems that may serve as a confounder and provide a worse prognosis compared to those who don't have underlying medical issues. Different histological subtypes, for example, diffuse type typically has a worse prognosis compared to intestinal. And similarly, specific biomarkers like deficient mismatch repair MSI high tend to have a better prognosis, say in the curative intent setting, whereas MET expression, or certainly gene amplification, tends to have a worse prognosis. The burden of disease, even within stage 4, if you have more cancer sites, larger lesions compared to those who have less cancer sites or smaller lesions, tend to have a worse prognosis. And so if you're doing a one-arm study without a control, and you just happen to enroll 60 patients, that was the target number to enroll to that study, but they all, or many of them, or a majority of them, tended to have good prognostic factors, it may seem that your study drug is really, really successful in terms of survival when compared to historical controls. But really, it may not be any better. It's just that you had patients that had a whole bunch of good prognostic factors, and that's why ultimately it looks like the outcome is better. So randomization to a control of standard therapy ensures that, or at least tries to ensure that there is an equal number of both good and bad prognostic factors in each treatment group, so that it eliminates this bias, patient selection bias. And now, if there is truly a benefit that's seen in your investigational arm with the new drug compared to the control arm, as well as being able to differentiate side effect profiles, now we can really say that it's from the drug and not from one of these underlying imbalances of prognostic factors. In addition to controlling for unknown or known confounders or prognostic indicators, random variation is an issue. And I'm going to take you through what is random variation. Let's take, for example, the flipping of a coin. We would expect that if you flip a coin, that you have a 50% chance of getting a head and 50% chance of getting a tail. So if you were to flip that coin 10 times, we would expect 5 heads and 5 tails. But is that always true? Well, the answer, of course, is no. Let's look at some examples. If you flip a coin 100 times, we expect the truth to see 50 times getting heads and 50 times getting tails, unless you have a trick coin. But if you have a regular coin, you should expect 50 heads and 50 tails, 50% chance of getting each. But would we always see 50-50? No. Sometimes we might see 49 heads and 51 tails. Sometimes we might see 53 heads and 47 tails. And even rarely, but sometimes, we might see 35 heads and 65 tails. And you can even go online these days and Google and find a calculator to calculate what's the likelihood of getting 65 tails if you flip a coin 100 times. And it's actually almost 9% chance. So almost 1 time out of 10 times that you were to try this as a study or a trial, you would get 65 tails. So even though we know that the true probability should be 50%. And so how is that possible? This is random variation at its finest. And so that said, what are the chances of getting 75 tails versus 25 heads? Well, that probability is really unlikely. It's still possible. Anything is possible, but it's like 0.0000019% chance that you would get 75 tails. What the concern here, though, is that if we didn't understand random variation, we might falsely conclude that we're more likely to get tails than heads, because look at 65 times it was tails and 35 times it was heads. So I think that this is a true association, whereas ultimately it's just random variation. And so the first thing we're learning, or the first principle, is that the larger the difference, the less likely that this would occur. And so you can bet that 90 tails versus 10 heads, although possible, would be extremely unlikely. And if that was to happen, we would actually falsely conclude that tails is more likely than getting heads in a coin, even though that was just a random fluke, random variation. The next principle to understand, though, is that what if we flip the coin instead of 100 times, but now we flip it 1,000 times? What is the chance of getting 350 heads and 650 tails, which is the same ratio as this one here when we flipped it 100 times, and it was an 8.6% chance of getting that. But now if you do it 1,000 times, it's a near 0% chance. It's not zero, but it's lots of decimal points later. And so the next principle to understand is that by repeatedly doing this, we will converge on the true reality, which is that the chance of getting a spurious value is less likely, and that whatever you do see is going to be real. And in this case, of where the result or the truth is that there's no difference between the probability of getting a heads or a tail. And by doing this 1,000 times, we've proven that it's a very unlikely to not get a very close to 50-50 ratio. So with hypothesis testing, with statistical analysis, this is what we are trying to do, is that we are assessing to see if there's an improvement in an investigational arm and whether it's real and try to remove the possibility that it is not due to mere random variation. And so in this example, what we're trying to eliminate is the risk of getting a false positive. So this would be a false positive. We would have concluded that this, without statistics, that this is more likely to be a tail than a head by flipping a coin. But with predefined statistical analysis, we know that this is actually within reason, that this is just random variation. In addition to ruling out false positives, saying a drug works when it doesn't, by doing a study with predefined statistics can also help to rule out false negatives, and to make sure that we identify a true improvement of a drug and not say that it doesn't work. So I'm going to show you now some examples of clinical trials that speak to each of these points. So first, why do we randomize? This is to get at ruling out patient selection as a reason for differences between outcomes. And so what we see here is a study that was called OEO2, esophageal cancer study. In Britain, they have an O in front of esophagus, so all the British studies are OEO. And so OEO2, the second study of this group, was looking at, in the curative intent setting, chemotherapy with cisplatin 5-FU for 2 cycles, then surgery, versus surgery alone. And this was the standard of care at the time. And this was published in 2002. Because it showed benefit, this became a new standard option. The next study that was designed was OEO5, looking at that new standard of care, cisplatin 5-FU for 2 cycles, versus an investigational arm of ECX, epirubicin, cisplatin, and capecitabine, the oral version of 5-FU, for 5 cycles. And this study was reported and published in 2017. And so what we see here over on the right are results from the study OEO2, showing the survival curve of cisplatin and 5-FU. Then we see the 2 arms of the OEO5 study showing the survival curve of cisplatin and 5-FU in blue and ECX in red. And so what we learned from this is that something called cross-trial comparisons, where we have a result from one study and we have a result from another study, and we're trying to compare outcomes across the different studies. And so we have come to learn that we have to be very careful when we do that, because in this particular example, if you were to compare the outcome of the cisplatin and 5-FU in 2002 versus the outcome of ECX in 2017, that's comparing the orange curve versus the red curve. The red curve looks clearly better. And if we weren't randomizing patients, this would have been declared a beneficial therapy and become a new standard option for patients. But if you compare the internal control arm, cisplatin and 5-FU, from the 2017 study versus the ECX, there's no benefit, and it's considered not statistically significant. And so how can this be that the standard therapy of cisplatin and 5-FU would be so different in these 2 different studies? Well, patient selection is a key. The differences in patient stages. Maybe there was more stage 2s versus stage 3s in that study. Maybe they had smaller tumors in the more recent study. Maybe they had more intestinal type versus diffuse type, just by random chance of the patients that happen to be enrolled. Certainly, it's also 15 years later or so, where different diagnostics were introduced. Diagnostic laparoscopy became more routine. PET scans became more routine. And so patients that would have been under the radar without these and considered locally advanced non-metastatic would now not be eligible for the OEO5 study. So we've excluded stage 4 patients. Surgical techniques have improved over the years. And so in addition to that, in the stage 4 setting, we've had improvements and advances in stage 4 therapy. So even in the stage 4 setting, patients live longer. And so for all of these reasons, the control arm, cis5-FU, is doing better than the cis5-FU from 15 years earlier. And so without this internal control, accounting for all of these and maybe unknown differences, we would have erroneously concluded that ECX was better than cisplatin 5-FU. And why do that? We would have been exposing patients to a triplet chemotherapy versus doublet that is more toxic, and that ultimately is not any better. So this is a classic example of why randomization is done to account for underlying prognostic factors that can be controlled for by randomization. Here's another example, Keynote 61, the name of the study. And this was a study that was done in the second line setting after failure of first line therapy, comparing pembrolizumab, a new anti-PD-1 antibody immunotherapy checkpoint inhibitor, compared to paclitaxel chemotherapy. At the time, we had already seen evidence that paclitaxel plus ramucirumab was better than paclitaxel alone in the Rainbow study. But this study used what many of us refer to as an inferior control arm of paclitaxel alone, partly because ramucirumab was not available in many countries around the world, and this was a global study. And so the argument was we are going to use what a global standard is with paclitaxel alone. And so why randomize though? Why assess subgroups, molecular subgroups in this case? Well, this was the result of the study of all the patients enrolled, hundreds of patients in each arm. And you can see the result of the control arm in the purple and the results of the pembrolizumab arm in green. And this was looking at any PD-L1 status. And just from this is now a classic crossing of the curves result, where it looks like some patients are doing worse and it looks like some patients are doing better. And so when analyzed to see, well, what if we look at it by patients who have tumors that are high versus low PD-L1 expression, we see this. Amongst those who had PD-L1 positive tumors defined by CPS score of 1 or higher, patients did better with pembrolizumab compared to paclitaxel monotherapy. On the other hand, patients that had a tumor PD-L1 CPS score of less than 1 actually did worse than what might be considered an inferior control arm, where remember, paclitaxel plus ramucirumab would look a little bit more to the right of what this purple curve even shows. And so without looking at these types of data, we may falsely conclude that there's no difference, because classically, when curves cross like this, it's thought that the therapy just doesn't work and we're just seeing random variation. But if we look at logical subgroups, in this case, PD-L1 expression for a PD-1 inhibitor, we see that, as might be expected, those that are expressing PD-L1 seem to derive benefit, whereas those that don't. This was a phenomenon that was seen over and over in multiple studies in multiple tumor types. Now, this is monotherapy pembrolizumab versus chemotherapy. What about when we look at chemotherapy with or without immunotherapy? So the immunotherapy added to chemotherapy, not replacing chemotherapy. And again, well, why randomize and why assess subgroups? Well, in this case, we saw a clear benefit of pembrolizumab plus chemotherapy versus chemotherapy alone in the first line setting in this Keynote 590 study, which accrued patients with esophageal and G junction cancers, including both adenocarcinomas and squamous cell tumors. And again, this is the survival curve, not taking into account the PD-L1 score. But if we do take into account the PD-L1 score, really what you can see here is that the patients who are deriving any benefit are patients that have high levels of PD-L1 compared to those who don't. Now, patients aren't doing worse than the control arm here, but that's because in this study, patients are getting immunotherapy plus chemotherapy. They're not just getting immunotherapy alone. And so this is why many of us really like to emphasize that PD-L1 is a good biomarker to help identify who will and who will not be likely to derive benefit from immune checkpoint inhibitors. So if we didn't randomize patients to a control arm of just chemotherapy alone, And if we didn't assess subgroups, we wouldn't be able to understand these principles. So the next example in question to show you is, why do a phase 2 study then? It seems to be wasting time and years in terms of getting the final result and getting access to therapy that might work. This is going to the zolbetuximab story and the phase 2b study, the randomized phase 2b study called the FAST study. And in that study, which again was recently published but conducted years ago, in the intention to treat group, all patients enrolled, which was defined as patients who had claudin expression of at least 2+ intensity on a scale from 0 to 3, and in at least 40% of the tumor cells in the sample. And you can see that there's a clear benefit with PFS, progression-free survival, and a clear benefit of overall survival. And the study design was, again, chemotherapy with epirubicin, oxaliplatin, and capecitabine, with or without zolbetuximab. So in this case, it was a positive study, and now we know in retrospect that the phase 3s are positive. Why was this step necessary? Well, one thing was, with phase 2 data, with drugs that are designed for a specific biomarker, like in this example, zolbetuximab for claudin 18.2 tumors, the cutoff of 40% of cells at 2+ intensity was sort of an estimate of maybe this will be a good cutoff to use. But from these data, maybe we can refine this and say what really we should be doing. And so this is a classic example where the study helped to understand who would actually derive benefit. And so amongst all the patients enrolled, they looked at a subgroup analysis of patients who had 70% of cell staining versus those that had less than 70 but more than 40%. And you can see that really the lower level expressors derive no benefit at all. There's no benefit between the two arms, whereas it's all driven by those at the higher cellular expression. And so, in fact, this is what led to the selection criteria cutoff for the phase 3 studies, and will now be the new standard way of looking at this. And so this was learned from this precursor phase 2b randomized study to really narrow down what that biomarker should be. So, of course, now we have the phase 3 study, which was reported, Spotlight study, which was using FOLFOX chemotherapy with or without zolbetuximab, and showing a clear benefit of the zolbetuximab over the control. A few things to point out is that this was a placebo-controlled study, and we're going to get to placebo and why we use that. So table that for now. But in addition to that, another principle is cross-trial comparison and why we randomize. And so this study showed a median overall survival of 18.23 months in the zolbetuximab arm. So median survival 18 months. If this wasn't a randomized study and we were comparing to historical controls where we expect median survival to be 11 to 12 months, we would have been overestimating the benefit of this drug. And so really not having the full picture. Whereas in the controlled study with a randomized control of just standard chemotherapy, we see that they seem to do exceedingly better than what we might even expect from a historical control of 15.54 months. And so why is that? It could be a number of factors, as we pointed out with the OEO studies, is that there could just been selection randomly of better prognostic patients for whatever reason. It could reflect to some degree better therapies that we have now compared to 10 years ago. There are some things that speak against that because more recent phase 3 studies are always showing 12, 13 months as a median survival in control arms. So another thing to mention is in biomarker selective studies like this one, where patients are newly diagnosed and they're waiting to enroll on this study. And one of the key eligibility criteria was that you needed to have tissue available to test for claudin and to be positive for claudin to be eligible. So patients had to wait until that result was finalized. And this could take weeks, if not months in some cases, to get that result. And so One thought is that patients who were sicker, who couldn't afford that time to be waiting around untreated 2 to 4 weeks after signing the consent form for screening for the study, would not be enrolled to the study. They would have been treated with standard therapy because they just needed therapy quicker. And so one other thought then is that we're selecting for patients that have better prognostic disease, less symptomatic, less advanced. And so both arms are doing better is the final conclusion. So this is again supporting the need of doing randomized studies to account for known or unknown prognostic factors and to really understand what the added benefit is of any therapy on top of a standard therapy. So in this case, the hazard ratio is 0.75, and the absolute benefit in terms of survival was an improvement in the median survival of about 2.7 months. So again, summarizing, randomization has this internal control to eliminate these biases. And so we can really best estimate what the benefit, or in some cases, the lack of benefit might be. So this is an example I've been taking you through phase 2 and phase 3 of the zolbetuximab success story. But as I've pointed out, there's probably more stories along the way that have unfortunately not been success stories. One example is the story of MET expression of oncogene that can be overexpressed in tumors. And a number of therapies were designed to inhibit this pathway, one of which was ralutumumab, which was an anti-HGF or hepatocyte growth factor ligand inhibitor, which is the red in this figure we talked about in the targeted therapies video. And so the idea being if we neutralize the ligand, then we would decrease signaling of MET, decrease oncogenic potential of the cells, and improve survival. And so why do a randomized phase 2 study? This was an example of a phase 2 study that was run of general chemotherapy with ibrutinib and cisplatin and capecitabine, which was an old standard of care for first-line therapy, with or without ralutumumab in a placebo-controlled fashion. And that study actually showed benefit of ralutumumab compared to the control arm. And so that said, what was also learned from this phase 2 study is that it really only benefited patients that had overexpression of MET. And so you can see here that patients who were MET negative, their tumors did not have overexpression of MET. There was really no difference. In fact, they did a little bit worse than the control arm, whereas ralutumumab seemed to improve over the control in the MET positive tumors. So this is what led to a phase 3 study for only MET positive tumors. So from a phase 2 study, the biomarker refinement story and concept is re-emphasized. But we also have to say that these were relatively small numbers. And so was this definitive? Did we need to do a phase 3 study? Absolutely, because these are just relatively few numbers. And we've learned that random variation can lead to these differences, not true differences in true benefit from the drug. And so that led to a randomized phase 3 global study called the RiloMET study, selecting only for MET positive tumors. And unfortunately, that study was negative. And in fact, the ralutumumab arm in red there had a worse survival compared to those that just got the standard of care. Why would this study be negative? We had a positive phase 2 study. Well, one idea might be that the biomarker selection might not have been adequate. 80% of tumors were being called overexpressed. So of the patients screened, 80% or so of them were considered positive. Maybe the cutoff was too lenient. Another concept is that the side effect profile of ralutumumab was relatively high and often overlapped some of the side effects of the chemotherapy drugs. And so naturally, during the conduct of the study, physicians would stop the chemo parts and keep the investigational agent on, and chemotherapy can be effective. And so by stopping effective chemotherapy earlier because of side effects, maybe that negatively affected the study arm with ralutumumab compared to the control arm. Ultimately, it could have been that there was random variation in that phase 2 study, and it was a false positive. We've talked about false positives and that they can occur. and that it looked better, but it actually truly wasn't. On the flip side, it could be that this phase 3 study is a false negative study. I think that's the least likely of all the explanations, but you can see the thought process that goes through the data that are obtained from observations from studies, and to try and figure out why things work, why things went wrong, and to maybe come up with strategies to address that in future studies. And so this circle of observing, hypothesis testing, repeating experiment, and going around in a circle can help pinpoint what studies might be effective and which ones may not be. Regardless, this speaks to why we wouldn't just say and declare that a drug works after a positive phase 2 study, because unfortunately there are a number of examples of this that the follow-up phase 3 studies end up being negative. And so again, with the intention to really be certain as we can be, that a therapy is effective or not, this type of process with scientific method and larger studies that are randomized are really necessary. So the scientific method then of repeating cycles of observation, and from those observations coming up with new ideas, new hypotheses, testing them through experimentation, clinical trials, and then going through that cycle over and over again, as well as independent verification. More studies independently run showing the same result are much more robust than just one single study. Some other semantics about clinical trials and statistics and hypothesis testing are that, say, for example, our hypothesis is that zolbetuximab treatment improves outcomes, survival, progression-free survival, overall survival, compared to not having it, is actually referred to as the alternative hypothesis. The null hypothesis would be that it does not improve overall survival. And so in the example of the Spotlight story, the primary endpoint was progression-free survival. And if that was met, then overall survival was tested and compared between the groups. And this is called a co-primary endpoint. And both of the comparisons for PFS and OS were hazard ratio comparisons. And with predefined statistics, we have to estimate what the control arm will do. And based on prior studies, that was approximately 11 months. And if our target improvement was a hazard ratio of 0.75, that translates into a median overall survival of 14.7 months in the treatment. And so from that, we can calculate how many patients we would need to perform a robust clinical trial. And so these pre-specified hypotheses really makes any observations and results robust. And so it's like thinking about Babe Ruth, who not only would hit home runs, he would point to where he was going to hit it, and he'd hit it there. And he really meant to do that. And so that goes a lot further than hitting the home run, seeing where it went, and saying, yep, I meant to do that. And so this is the whole concept, statistics and hypothesis testing, is that we are pre-specifying what's going to happen. And if it happens the way that we anticipate that and hope that it does, then it makes it much more believable and statistically sound. So I was alluding to numbers calculations. How do we know how many patients should be enrolled in a study? Why is it 587 patients that need to be enrolled? And the reason that we do this, first, is to provide some certainty that the result is not a false positive. That's referred to as the alpha level. And secondly, we want to provide some certainty called the power of the study, or the beta, to identify a true benefit, limit false negatives. We don't want to miss the truth and say that it's negative, even though it is actually beneficial. And the next thing we have to understand is that phase 3 alpha and beta levels are different than phase 2 levels. And so in a phase 3 study, where we really want to limit false negatives and false positives, we make the analysis stringent. And so we usually set that p-value, or the alpha level, at 0.05. What does that mean? That means that if you see a result and it is showing that your investigational therapy is better than your control arm, and the p-value is 0.05, then that means that it's a 5% chance that the result is positive. In other words, if you ran this study 20 times, one of the 20 times it would have been positive, but it's not really positive. And you would be declaring this, if you were to just base it on that one study, a positive result, even though it's not. That said, 19 of the times it would be positive. And so uh, that was arbitrarily set as a consensus to say that is a reasonable false positive rate. That, if it's showing a result and it looks positive, but it could be a false positive, but it's only a 5% chance that that's the case, then that's reasonable. The beta level of 0.1 means that you have a 90% chance to identify a true positive. So if it's negative, there's a 10% chance that it actually is beneficial, but you're declaring that it's negative. And so again, that was accepted as a reasonable cut point of false negative rate. On the other hand, phase 2 studies, which remember are sort of like the gatekeeper studies, are a little bit more lenient in terms of the false positive rate, where they allow for a 10% false positive rate. And on the flip side, they allow for a 20% false negative rate. And so why compromise like this? Well, a principle that has been coming up along the way is that you need a lot of patience to find small differences and to be limiting false positive and false negative rates. Whereas you need fewer patients to be enrolled to a study to get a little bit lower thresholds. And so as a consequence of doing smaller phase 2 studies, we understand, and there's a trade-off, that while the benefits are that we can get the study done quicker, we can get results faster, there is more risk that those results would be false positive or false negative. Phase 3 studies, which again, there's a reason why they're considered more definitive, is because they're larger and that they limit the false positive and false negative rate. And with the results that we see can be better accepted as the authoritative result. So again, principles that we need to understand are that we need more patients in a study if we expect to see a smaller difference. And we need fewer patients in a study if we expect to see a larger difference. We don't need as many patients if we expect the result to be showing such a large magnitude of benefit with the investigational therapy versus the control therapy. So let's look at the example then of the zolbetuximab spotlight study, which again, the primary endpoint was progression-free survival and overall survival. And the secondary endpoints were other clinical benefits like response rate, disease control rate, and of course, chronicling the side effects. And the numbers calculation of that study showed that we needed about 550 patients enrolled to show both the PFS and OS co-primary endpoints. So how did we come up with this number calculation? So the numbers needed of 550 patients were derived by starting with what is the control arm estimate median survival, what is our anticipated target median survival in the investigational arm, what's the target hazard ratio, and from that, stipulating what the alpha and the beta, the power, are going to be with these base parameters. That's how the numbers of patients to be enrolled to the study was derived. And so every study has these specific calculations pre-hand that then after the fact, if the result is observed and it makes the benchmark, then it's considered a positive study. If it doesn't, then it's considered a negative study. So a little bit more about detecting the magnitude of benefit. Remember the coin experiment of flipping the coin and what the chances were of getting 65 versus 35, but the same ratio at much higher repetition, and the chances of that, they changed because the more times you do it, the less likely you'll get a false positive. But the same is true for eliminating false negative rates. The higher the repetition or the more patients enrolled in a study, the less likely you'll get a false negative rate. And so a classic example is looking at the Keynote 62 study. This was a study in the first line setting for gastric cancer, looking at chemotherapy with or without pembrolizumab, Keytruda, immunotherapy checkpoint inhibitor. And this study, while it showed a trend to benefit of pembrolizumab and chemotherapy, it was considered not statistically significant. The hazard ratio was 0.85, but the confidence interval and the p-values suggested that it was random variation. It was crossing the boundary of one, and the p-value was not less than 0.05. And you can see the absolute difference in the median survivals was 1.4 months in favor of pembrolizumab compared to no chemotherapy. It should be noted that this was in patients that had a PDL1 score of CPS 1 or higher. Well, a similar study called Checkmate 649, looking at a different checkpoint inhibitor, nivolumab, Opdivo, was run. And it should be known that, generally speaking, physicians who use these drugs consider these drugs interchangeable, relatively equivalent. Nivolumab, pembrolizumab, and almost every company has one, and they all show very similar benefits. So this study with nivolumab was enrolling patients with chemotherapy with or without nivolumab. And this study was showing an improvement in absolute benefit of 1.8 months in favor of nivolumab, not much different than pembrolizumab, but its p-value was considered statistically significant, less than 0.05, and its hazard ratio confidence interval was not crossing one. And so this study is considered positive, and this study is considered negative. What's the difference? I mean, we all understood that these therapies were relatively similar, and the chemotherapy backbones were not that dissimilar. They were 5-FU with platinum backbone. So why? Well, one glaringly obvious difference was the numbers of patients in each of these studies. This Checkmate 649 study was more than 3 times the size in terms of numbers of patients enrolled. And so remember the principle that if it's a smaller benefit, here we're talking about an absolute benefit of less than a couple of months, the more patients, the more likely you'll detect that and eliminate a false negative result. And speaking to that, the follow-up study of pembrolizumab called Keynote 859, which basically ran the same study again, but with larger numbers, 3 times the size of Keynote 62, this study was positive and essentially eliminated a false negative result. So understanding these principles helps us understand why a study like Keynote 62 is considered negative, while Checkmate 649 is considered positive. Both studies were otherwise very similar in terms of biomarker assessment. Both were enrolling only PDL1, CPS1 or higher. Both had a checkpoint inhibitor with or without chemo. And so it didn't make sense that one was negative, one was positive. But as you see, it really came down to the size of the study, finding smaller differences. We've been alluding to endpoints in clinical trials and in the statistics and hypothesis testing. And whether it's a stage 4 study or a locally advanced study, there will always be a pre-specified primary endpoint and then a number of secondary endpoints. The primary endpoint is the prime statistical analysis is based upon. And ultimately, for all of us, the patients, the physicians, we want to improve survival. Survival is considered the most robust endpoint in terms of oncology studies. However, it takes a little bit longer to observe that result compared to an earlier endpoint. Earlier endpoints, also called surrogate endpoints, are endpoints that may help predict the overall survival. And so examples of those would be progression-free survival, how long the therapy works while it's being given. In the curative setting, it would be disease-free survival, how long a patient doesn't have any evidence of cancer after curative intent therapy. Response rates of the treatment can be assessed relatively early. Within the first couple of months, you can see, is it working or not? And some tests I've alluded to in our liquid biopsy video, like a blood ctDNA analysis, can assess whether it's going to respond within a few weeks of therapy. So these surrogates which tend to be sooner, don't 100% predict overall survival. And so that's the trade-off, is that you can use earlier endpoints, but then your survival estimate may not be as accurate. And so the advantage, though, is that with an earlier analysis, you need less patients and you can get earlier looks at the data. So these types of surrogate endpoints are often used in phase 1 and phase 2 studies, whereas OS and PFS, OS and DFS are used in larger, more definitive phase 3 studies. Now we're going to move to why we use placebos. From a patient perspective, I can certainly understand why both randomization and or placebos are really off-putting. You want to get a potentially new therapy. But what I'm trying to point out as we go through this is that there's a reason why we do randomization, because ultimately we could be declaring a therapy beneficial that's not. In some cases, there are examples where you could declare a therapy not working well, but it is, but you didn't have an appropriate control. And so at the time, for patients enrolled in those studies, of course, only a subgroup of the patients are getting the therapy and a subgroup are not. But later on, patients years from now will be benefiting from studies that are being accrued to today, just like patients being treated with standard therapies today are benefiting from patients who enrolled in studies previously. So getting to the placebo, which is a blinded study, why do we do that? Using a placebo in the control arm is required if we want to really control for biases. We've talked about many biases, but randomization doesn't necessarily exclude all of these biases. For example, physician bias. If it's not a blinded study, the physician knows who is and who isn't getting the drug of interest. Physicians want the drugs to succeed also. And so whether consciously or not, decisions are being made in terms of when to stop therapy based on scan results, based on patient symptoms and side effects. And so if it's known who is and who isn't on an investigational therapy, decisions can be made and swayed in favor of the investigational drug, again, whether it's consciously or not. And similarly, there's a known patient bias. If a patient knows they're on an investigational drug, they also want the therapy to work. And so they may report better outcomes, less symptoms of pain, less cancer-related symptoms, improved quality of life. And so this is a well-known, established fact, the placebo effect. And so if patient and physician don't know, then we've excluded that source of bias. And so a double-blinded study means neither the physician nor the patient know if they're on an investigation drug or the placebo. And so really, if there's still a benefit that is observed at the end of the study when the results are analyzed, then one can really feel confident that any differences are truly and solely due to the therapy that is added to the standard and not having to do with any biases that now have been limited by randomization and a placebo control. This is a really important thing to say because this is a huge concern of patients, understandably. But the placebo arm of any study will always be getting a standard of care plus the placebo and never be deprived of known effective treatments for a study. This is why they go through institutional review boards, IRB committees, et cetera, is that the control arm always has to meet the standard of what is being done anyway. And so this should put many people's concerns to rest with a placebo-controlled study, is that they're always going to be getting standard of care. And that they may be getting, in addition to that, an investigational drug that may or may not be beneficial. We don't know the answer to that because the study hasn't been done yet. And that's why the study is being done in the first place. So then the next thought in a patient's mind might be, well, why would I want to do this then? I mean, I have a 50% chance of getting the drug and 50% chance of getting a placebo. And why would I want to do that? Well, the response to that would be, well, you have a 0% chance of getting the drug otherwise. And so this is the only way to get the drug right now. And the other response would be is that we don't really know that it works, even though we want it to work, even though there's evidence preclinically in earlier phase studies that suggest it works. I've been taking you through reasons why that's not necessarily true, that it will ultimately truly work. And only in the study like this will we understand and know for sure. The other thing to say is that I mentioned that there'd be a 50% chance of getting the drug and a 50% chance of not getting the drug and getting a placebo. In recent years, because of the understanding that we know that patients really would prefer not to have a placebo, is that the ratio of the numbers of patients getting an investigational drug versus placebo can be changed. And so, for example, some studies enroll 2 to 1 in favor of the investigational drug to placebo. So for every 3 patients enrolled, 2 would get the investigational drug and 1 would get placebo. And so that is trying to stack the deck in favor of getting the therapy and giving you a better chance of getting the therapy. But at the same time, it still has a placebo arm so that we can control for all of these biases that we've been talking about. Why placebo? So this is an example of a study to highlight why we use placebos. This is a study called the REGARD study, which was a second-line study in the era where second-line therapy wasn't a global standard to use. So after first-line therapy, the standard option was best supportive care. And so the control arm was placebo alone. And the investigational arm was ramucirumab, an anti-VEGF receptor 2 antibody. And this was the precursor study of the RAINBOW study, which was looking at paclitaxel with or without ramucirumab, which is now a standard of care in second line. But this was the precursor study. And so it was ramucirumab monotherapy versus placebo without any chemotherapy. And what we saw was on the left, overall survival was improved with ramucirumab versus placebo, and PFS was improved, albeit modestly for both. Why do I show you this? Well, the response rates. There were 3% of patients enrolled in the placebo group, getting nothing else, no other chemo, no other treatments, that had official objective response. And so this is an example of the limitations of a lot of our measures, these surrogate endpoints that I've been talking about, particularly response rate, compared to, say, overall survival, where CT scans have subjectivity in them, and especially if it's controlled by placebo, where we don't know if a patient is or isn't getting a therapy, and we're just left with the scan results, some patients' cancers may look smaller from one scan to the next, and for various reasons. But the fact and the point is that even placebos can show response. Now, it's not many, but it is a number. And in fact, in this particular study, the response rates were no different with ramucirumab alone versus best supportive care. So the next question one might ask is, how does this drug work to improve survival and if it's not shrinking the cancer? Well, the disease control rate was better. And so it was able to control the cancer longer than without it. And so ultimately it showed that that translated into a progressive free survival and an overall survival and led to its approval as a monotherapy. In addition to that, placebos and randomization really help us to look at the differences in side effects of an investigational drug versus a control. And so in this case, you can see that there were some specific side effects that were higher in the investigational ramucirumab arm. Classically, anti-VEGF, anti-angiogenesis drugs increase blood pressure and increase slightly the risk for bleeding. And we see that. But at the same time, if you weren't randomizing, you may blame a lot of side effects on a drug that really aren't from the drug at all. The placebo arm had similar rates of fatigue, appetite changes, vomiting, constipation. And ultimately, these problems were more likely due to the cancer itself rather than the therapy because the rates of these were similar across the arms. So by Comparing with randomized and placebo controls, we can really assess not only outcomes, clinical benefit, but also compare side effects. And so when patients come to me now and ask, well, what are the risks and benefits of any given drug? I can tell you because we have evidence that has shown us these results robustly. So we've gone then through the painstakingly long trajectory from preclinical through the phases until ultimately a drug can be approved as a standard therapy. This next little segment is to talk about when drugs are showing such remarkable outcomes in early phase studies, and they can leapfrog across some of these steps if they're just obviously showing such dramatic benefit. What examples are there? Well, there's many across oncology, but the one for gastroesophageal cancer really is looking at pembrolizumab for mismatch repair tumors, MSI high tumors. And the data were becoming so obvious, even with small numbers of patients enrolled to even uncontrolled studies, that it was just so obvious. And so I'll show you the data in the next slide, but this led to an accelerated approval in 2017 based on relatively small numbers of patients and also in an unprecedented strategy by looking at different tumor types in the same study, where classically and even today, studies are really limited to a given cancer type, gastric esophageal, or only breast, or only lung. Whereas this study didn't care what type of tumor it had, as long as it had mismatch repair MSI high tumors. And so from the original study, looking at really small numbers of patients, relatively speaking, 86 patients, of which only 5 had gastroesophageal adenocarcinoma. You can see that here, that gastroesophageal cancer is made up a subset of all the patients enrolled. But the results were so striking because this study was in heavily pre-treated patients, had failed all of standard therapies, and was having a waterfall plot. Remember, a waterfall plot is looking at the response rate. And so each bar is a patient and each color is the different kind of cancer. And so you can see that the gastroesophageal cancer patients, you can see that a large proportion of the patients are having responses, and a lot of them are having complete responses, 100% complete response of the cancer visible on scans. And with longer follow-up, even more depth of response, more patients having responses. And so this was really remarkable. And to put in perspective, the response rate of chemotherapy in first line is around 40 to 45% of patients, say with FOLFOX, which will have shrinkage of their cancer. This was in late line, failure of all therapies, where typically therapy responses decrease with each line of therapy. And we were seeing a 60% response rate in gastroesophageal cancer. It's only 5 patients. Could this be random variation? Sure. But for all of these patients lumped together, a response rate like this to be random variation would be highly, highly unlikely. And in addition to that, we see that the progression-free survival and overall survival was showing outcomes that looked as if the patients had had surgery with curative intent, where curative intent curves usually drop a little bit, but then plateau. And the patients below that are cured. And whereas in the stage 4 setting, typically we see the curves continue to fall downward. And so not only were we seeing remarkable responses, but some of these responses were durable. And so when this was seen, this was a prime example where why would we put such a drug through all the phase 2, et cetera, studies when it's so obvious that this drug is a home run with patients years out, alive and without cancer and off therapy, because many of these studies early on with checkpoint inhibitors require that you stop therapy after 2 years and just follow it. And so off therapy, no evidence of any cancer anymore. It was obvious. And so with follow-up of more patients accrued with this strategy, and compared to a historical control, not even randomized, not placebo controlled, it led to a pan tumor, any tumor type that had MSI high or mismatch repair deficiency in the second line setting or higher was eligible to get pembrolizumab. This is an example of where if the results are so obvious and the benefits are so large that we will use logic reasoning to make a quicker approval. I will point out, though, that until just recently, these types of immunotherapy drugs, even for MSI high tumors, were not available in other countries, despite these data. And so they were still working with the framework of more traditional studies to really rule out false positive, false negatives. But again, the chances that this were false positive results being so strikingly beneficial was extremely low. And now more and more evidence supports this original decision years ago. Okay, now finally, we're going to get to some logistic things to consider when talking about clinical trials. Clinical trial eligibility. I mentioned that along the way here. Clinical trials have a protocol that stipulate who's eligible for the study and who's not eligible. These are called eligibility criteria. And to be eligible for a study, one has to meet all of these criteria. And this evaluation to ensure you're eligible happens initially when you're first seen by the physician and their team at the time of a clinic visit to make sure that the obvious things are met. But after you review the protocol and sign consent, that's when a more in-depth analysis to really see if you're eligible on every criteria that's stipulated is there. And so I always would warn patients that, yes, we can sign consent to screen for this study. Screening means we're going to make sure that you're eligible based on all the details and all the fine print to make sure you meet all the eligibility criteria. And it may be that there may be one that you don't meet, and ultimately, even though you sign consent, you're not eligible. And so examples of eligibility criteria include lab values, are pretty routine. For example, white blood cells have to be at a certain level or higher. Tumor histology, it has to be a stomach cancer or an esophageal cancer that's adenocarcinoma. Biomarker, it has to be HER2 negative to be eligible, or it has to be HER2 negative and claudin positive for, say, the zolbetuximab study. Sometimes patients that have peritoneal ascites aren't eligible for studies, for example. The stage, of course, has to be a certain stage. Usually it's metastatic stage 4 or locally advanced has a stage 2 or 3 to be eligible. A common one, especially in later line studies, especially in non-randomized studies looking at an endpoint like response rate or progression-free survival. One needs to have measurable disease on a CAT scan so that we can follow for response or not. Talked about in many of the videos, like say peritoneal only cancer, which is common in gastroesophageal cancer, where we know it's there, but we can't see it on scans. And so that would not be considered measurable disease. And unfortunately, and frustratingly, many patients with this subset of gastroesophageal cancer aren't eligible for trials because of this criteria. And the definition of measurable on CAT scan is that a nodule or a cancer spot has to be at least 1 centimeters in size or bigger. And lymph nodes have to be 1.5 centimeters in their shortest dimension because they give a 2-dimensional size. And so it has to be 1.5 centimeters in the smallest dimension. And so many patients who I know have cancer, and I've been treating them for a while, and their previous therapy had stopped working, but they don't have measurable disease, would not be eligible for many studies and many studies that include this particular criteria, something to be aware of. Another big one is the line of therapy. And so when we talk about line of therapy, what does that mean? That means, first line refers to the first therapy used in the newly diagnosed stage 4 setting. Second line would be the second one used after the first one either stopped working or is not tolerable. Third line, etc. And so usually clinical trials stipulate you have to have cancer of this type in this line of therapy. And so a third line study would say you must have received 2 prior lines of therapy, for example. Newly diagnosed, never treated before would be a first line study. And something to be really understanding is that even 1 dose of other therapy, standard therapy, would then make you ineligible for a study that's in the newly diagnosed setting that requires no prior therapy. So this is another frustrating thing where patients are seen at a center where there's studies and they've already started therapy, locally, for example, they've had a dose of treatment 4 days ago. And so, unfortunately, when we were considering studies, they would not be eligible for a first line study that requires no prior therapy. So this is an important thing to understand. And when we were talking about the video on second opinions, is that it's nice to get a few opinions before we start any therapy if we can afford to. One of the other challenges is that many patients have a lot of symptoms when they're first diagnosed, and they just need to get therapy started as soon as possible and can't afford to wait. And that's in that circumstance, there's nothing more that one can do. But if one is feeling okay enough to go and get another opinion and screen for a study if one is eligible, usually this will delay the onset of getting therapy started a couple of weeks on average, but ultimately may allow you to get access to new therapies. What about the duration of therapy? Usually it's stipulated in the protocol that it's used until the cancer learns how to grow on it, or called disease progression. And so that said, there are examples of extreme responders. And I showed you the MSI high mismatch repair data with pembrolizumab, where a number of patients were having complete responses still 2 years after starting therapy. And in those cases, that particular protocol indicated that we stop the immunotherapy treatment and just follow. And so every study is written a little bit different. Some are 12 months later, et cetera. Some say you stop, but then you have the option to re-challenge if and when the cancer was to recur. And so all of these things need to be taken into account. And this is talking about the subset of patients on a study that might have this long durability of treatment. For the most part, these treatments are expected to work for a period of time and then stop working. And so most people would fall into the category of until disease progression. What about where the studies are taking place? The location of the treatment. Because there's a lot involved with clinical trials in terms of data collection, laboratory draws, research studies, et cetera, they're required to be given at a certain center where the study is being conducted. The uniformity of treatment is desired to limit variation. Treatments are required to be given, therefore, at the enrolling site and not really locally where you live. And so this is commonly at an academic center, which may be some ways away from you, which obviously isn't as convenient, but it is a way to get access to newer therapies. Fortunately, there are some studies that local community practices can participate in as satellites of larger studies like phase 2 and phase 3 studies, part of those cooperative groups, part of academic satellite centers, which is a phenomenon going on across the country. And even pharma-sponsored studies go directly to private practices. And so another thing to note, and why I wrote phase 2 and 3 studies, is that phase 1 studies are really involved in terms of multiple lab draws, even in the same day, staying for hours to get these to show the drug levels, because remember, these are often first in human, trying to look at as much detail as possible. And so these are really done at specialized centers like academic centers. And so they need to be done at those centers. Finally, a common question about clinical trials is this going to cost anything to me more than usual? And the answer to that typically is all the standard types of visits, treatments, labs, scans are all billed and done the same way to insurance. If it's considered routine standard care, it's billed to insurance. And at the same time, if there's any research tests, research blood draws, research procedures, and of course the drug of interest of itself in a therapeutic study will all be covered as part of the study and not billed to your insurance. So this is a common concern, but ultimately the answer is that it shouldn't cost any more to you or your insurance than standard of care, because anything that's considered research is covered by the study, and anything that's considered standard is just billed the routine way. So clinical trials then have led to incremental improvements over time. I've shown you the data that improved best supportive care with monotherapy chemo to doublet chemotherapy. And I've shown you some examples of targeted therapies for targeted populations and the median survivals that are slowly incrementally improving. And ultimately, of course, studies being done today are trying to improve on those. And an example is another tumor type of a goal, so to speak, is that colon cancer, that stage 4 historically had similar survival rates. And now with, again, this incremental strategy, has a median survival in some subtypes of colon cancer of about 3 years. And so this is the continual progress that we seek by doing clinical trials and learning which agents work and which ones don't, which patients they work for, which ones they don't, over time, so that at any given time point, the standard of care is actually robust based on our knowledge from prior clinical trials. In summary, then, clinical trials are used to determine how we can advance our understanding, how to best treat our patients. It's the only way, though, to scientifically determine with statistical scrutiny that the new therapy is actually and indeed safe, beneficial, and worthwhile compared to an already available standard therapy. Why expose patients to a drug that's expensive with toxicity and side effects if we prove that it doesn't work any better than current practice? If we don't do those studies, if we didn't do a randomized OE05 study, we would have inaccurately concluded that this looks better than historical controls. Let's just use ECX. Whereas we learned that with an appropriate study design, that it actually was no better. And we could spare future patients this side effect profile. So this clinical trial is a safeguard to prevent new agents from becoming standard that actually don't work, but just cause side effects and cost a lot of money. In addition, these studies can help us to really systematically chronicle all adverse events, side effects, and toxicities, so we understand those safety profiles, so we can explain that to patients, that we know what the potential benefits are, we know what the potential risks are, and now we can make an informed decision as to whether or not we want to proceed with this given therapy. This is ultimately what differentiates Western medicine from various products that you can find on Google or you've heard through the grapevine, is that those things are not rigorously tested. And the best we can do as physicians is say, when asked about these, is we don't know if they're going to work or not, and we don't know what their toxicity profiles are going to be. Whereas I can tell you with pretty good certainty what the potential benefits and risks are of this therapy because it's been rigorously tested. And so this alternative medicine, which we'll have a video for in the future, is alternative to Western medicine, which has been studied, and we understand the risks and benefits of any given therapy that works, and we also know things that don't work based on studies. Whereas alternative medicine basically is things that haven't been tested, and people can claim they work all they like, but we don't have evidence to suggest that's the case. And so it's not to say that in the end, if it was tested, that it did work. It's just saying that we don't know because it's not been tested prospectively in a clinical trial setting. So I'm in the camp that I'm not opposed to alternative medicine per se, other than to say you should discuss that with your physician, what you're thinking of taking, and to make sure that there isn't a potential interaction with what you're thinking of taking, some herbal therapy, for example, with the actual therapy that we're giving, because there could be negative interactions. There could be negative interactions that we know could happen, but also there could be interactions that we don't know because it's not been studied. And so my advice usually is I'm fine with doing some sort of alternative strategy during a treatment break, because when we get to therapy in the palliative setting, we'll talk about strategies of therapy, and that treatment breaks, treatment holidays are common in at least in my practice. And so during those breaks, that's the opportune time to try other things if you want to, because then at least we're not going to have a negative interaction. And so something to just consider, because ultimately, again, we'll end with that a physician's priority is to first do no harm. If we don't have enough information on a given treatment to be able to assess potential risks and potential benefits, then we may be actually causing more harm. And that's something that we always want to avoid as much as possible. So in this video, I took you through the principles of clinical trials from the perspective and examples of gastroesophageal cancer, looking at the design of clinical trials, pre-specified hypotheses, statistics in order to try and eliminate false positives, false negatives, randomization and placebo to eliminate biases and to really tease out what the benefits, if any, are of an investigational therapy, and also compare added side effect profiles compared to the control arm. And so I showed you a number of examples of clinical trials, why we use randomization, why we look at placebo, and we looked at the intentions and designs of phase 1, 2, and 3 studies. And finally, I went through some logistics in terms of common questions about clinical trials, the duration of therapy, the cost of therapy, the location of therapy. Am I eligible for studies? And the last thing I'll say is a common question is, how do I find studies and how do I know if it's a good study for me? This is where the second opinions and getting evaluated at academic centers where clinical trials are being run is really important to get an assessment of whether you're eligible for studies. So from there, you'll know what study options you have, and then you can decide with the assistance of your treating physician and also the physician running the study whether that study is a good fit for you. And so I'll end with saying that in oncology, there's always room for improvement in any situation, whether it's the curative setting or in the stage 4 palliative setting, where we have standard therapies which have been shown to be beneficial in previous studies and why they're currently the standard. But as you can see from outcome data, there's always room for improvement, and clinical trials are designed to try and find ways to improve on those standards. So patients should be encouraged in any setting to consider clinical trials if there's one available, if it's the right fit for the patient and their goals.